Recording in progress.
Treat inflammatory bowel disease, and in some ways it's related to the paper we talked about on Tuesday that had to do with pulmonary artery hypertension, at least the component of it that's associated with an inflammatory response. And so we'll be taking a look at a drug which I received on a notice had recently been approved, but in looking at it closer, this drug has been available since the mid-2010s. And what it was recently, the approval came for it to be administered subcutaneously as opposed to an intravenous infusion. But the drug has been around for a while. It was initially developed to treat inflammatory bowel disease and was one of the first drugs in its class that had been approved. It's since also been approved to treat other diseases associated with inflammation, especially autoimmune diseases such as arthritis, as an example. So I thought we'd take a closer look at this particular drug because it represents a class of drugs that have really become very popular for treating things such as psoriasis, arthritis, Crohn's disease, ulcerative colitis, and such. So let's take a closer look at this drug from the standpoint of its mechanism. Okay, hold on here just a moment, and we'll do a share screen. Hold on just a moment. Okay. So can you see the paper? All right, so that's sort of where we are. So the drug is infliximab, which was, as I say, developed in the approved in the late 1990s, but really approved for use in the 2000s. And the way it works is it is an antibody against tumor necrosis factor. So the thing that's interesting, I think, about this drug is that it is an antibody which is essentially a chimera between murine or mouse variable fraction of the antibody fused with a human component, which is the constant region of the antibody. And I'm going to throw a question out there to you. Why? Why have a monoclonal antibody that is a fusion between a murine and a human antibody? What do you think? Why would that be?
So, Dr. Johnson, over here appears like the FC region is humanized because basically the researchers don't want some of the antibody reaction against the drug itself.
Right.
Like they don't want to generate drug like antibodies against the drug, basically, and that is why they wanted to keep the FC region humanized so that it does not trigger an anti-infliximab reaction.
Okay, very good. I guess the question is then, do you think it'd be possible to develop a totally human antibody to TNF? Maybe not. I mean, TNF is a human protein. It might be difficult to generate an antibody to it, where certainly it was a murine antibody, you could certainly generate a
So Dr. Johnson, I fear like if we generate like 100% humanized antibody, possibly it may lack antigenicity, like it may not serve the purpose.
Oh, very good. In reading through the paper, though, one of the adverse effects to the drug is the development of antibodies to it. So having the chimera with the constant portion being human doesn't totally eliminate an antigen response to the antibody, but it seems to be fairly effective. Now, they sort of state in the paper that this antibody to TNF, so it decreases TNF. What's the role in tumor necrosis factor? What's the general role of TNF? Anyone want to kick in? Well, Vivek is very responsive, so we'll give him a break. Anyone else want to share? What stimulates TNF?
So in the article, TNA can activate the NF-κB and other signaling like a MAP kinase to like regulate the immune cell activation or like other functions like migration, something like that.
Well, that's true. TNF can activate all kinds of things, like the MAP kinase pathway and other pathways. But what triggers TNF to activate that cycle? Any thoughts? I mean, you guys are all on computer. If you want, why don't you take a few moments to do a quick online search for TNF and what it does. Why don't you take a few moments with that?
It's a regulator of inflammatory response.
Well, that's right. And what triggers it? Be triggered by infections, right? It can also be triggered by other stressors that will, normally trigger an inflammatory response. Typically, inflammatory responses occur with any kind of tissue damage, will trigger a response, and especially with infections that can involve TNF alpha. TNF alpha then, as Young mentioned, can trigger the MAP kinase cascade, but what other cascades can it activate, or what other mediator of inflammation does it play a role in that they sort of focus on in this paper?
Also, JNK and other pathways are caspase as well.
Right. By the way, what is caspase involved in?
Apoptosis.
Apoptosis, that's right. Does apoptosis play a role in inflammatory bowel disease? Well, the answer is yes, right? The nature of inflammatory bowel disease involves damage to the epithelial lining of the intestine with Crohn's disease and the lower intestine with ulcerative colitis. And so as part of that, the epithelial lining of the GI tract is triggered to have a certain level of apoptosis, which then allows all these mediators of inflammation to go in. Ultimately, you have this inflammatory response in the lining of the bowel, which can lead in ulcerative colitis to actually hemorrhaging. You could also have the development of fissures, which then can lead to ultimately fistulas and just leakage of intestinal contents into peritoneal cavity, which is of course a bad thing, because that then can lead to peritonitis, which is frequently a fatal infection of the peritoneal cavity. So apoptosis of the inner lining of the GI tract is a part of that. There is another mediator, though, that I want to talk about here in just a moment, which is NF-kappa B. And hold on just a second. I'd like to bring up another figure. Hold on just a moment. So I'm going to stop this, and we're going to show something else. So what this figure shows is the pathway of activation of NF-kappa B, which is essentially found in many cell types. And so you have an exogenous molecule, and this could very well be the receptor for what we've been talking about, just in terms of tissue necrosis factor, can bind to its receptor. Once it does that, activation of these receptors then activates this kinase, and what that kinase then will do is, of course, kinases do a phosphorylation, but they do a phosphorylation of this I-kappa B protein that then causes it to dissociate from NF-kappa B, which is a dimer, a heterodimer. The heterodimer then leaves the cytoplasm and enters the nuclear material, enters the nucleus, where it binds to a nuclear receptor after attracting coactivators and RNA polymerase, which then activates genes that are responsible for the synthesis of inflammatory proteins. The mRNA from those genes then go to the cytoplasm again, where they interact with ribosomes to generate proteins that are inflammatory proteins that then can be released and interact with typically different components of the immune system to develop an inflammatory response. So in terms of NF-kappa B, also plays an important role in promoting inflammatory factors. And so to the extent that infliximab inhibits tissue necrosis factor, it also then inhibits this TNF kappa B which then inhibits the synthesis of these other inflammatory proteins. Does anyone have any questions on that? Okay. Well, I'm going to go back to the paper, so hold on just a moment. Okay, so infliximab is essentially an antibody that inactivates tumor necrosis factor, which then will decrease a number of essentially decrease other mediators of inflammation. So let's take a look at those mediators. Well, but first, if we look at number 2 here, you can see that the drug can be effective in decreasing the symptoms of Crohn's disease and promoting relapse. By the way, to the extent that inflammatory bowel disease essentially becomes an autoimmune disease, the pattern of autoimmune diseases without treatment is to have periods of disease followed by spontaneous remission followed by relapse. And you see that with inflammatory bowel disease, you see that with rheumatoid arthritis, you see that with MS, multiple sclerosis, and other autoimmune diseases. Trying to think of myasthenia gravis, another autoimmune disease. And so in response to treatment with this TNF antibody, you see a decrease in symptoms, and they go through various studies here that look at clinical responses, where, as an example here, let me just find one. Here, I'm going to look at this one. Well, if we look there, you see for, let's say, a 5 or 10 milligram dose at 4 weeks, 22 of 27 patients had a clinical response, compared with only 4 of 24 in the placebo group for the comparison, or 33% went into remission compared with only 4% of patients. What does this say? Does this antibody against TNF work in 100% of cases? The answer is no, it doesn't. It only works in a fraction of cases, and that brings up the question of why. If this, in fact, is going after TNF and at least shutting down that one pathway in terms of NF-kappa B, why isn't it more effective than it is? What do you think?
Sir, maybe there are more other inflammatory mediators which are causing inflammation and That's why it's more related with the other like other than the TNF alpha inflammatory mediators which are causing the disease rather than TNF alpha. The effect of TNF alpha is only inhibited by infliximab.
Well, that's right. I think, was it you, Melanie, who mentioned MAP kinase? Or is that someone else?
Yes, yeah.
Yeah. So that's another signal transduction pathway. The other thing that's clear, and I am confident that Dr. Kavanaugh went into this in some detail. Did she talk about crosstalk?
Yeah.
Yeah, where activation of one transduction pathway can result in the activation of other transduction pathways, which then can produce a result. Now, in the overall realm of things, I think I may have mentioned at one time or another, with any kind of critical function in the body always has almost, I'll call them backup systems for redundancy in terms of getting the function accomplished. You could see this in the cardiovascular system. You could see this especially when it comes to eating. All key respiration, all functions that are life-sustaining rely on more than one regulatory system. And I suspect in the case of this autoimmune disease with the immune system, as you know, the immune system, at least in my thinking, is one of the most complex systems in the body, just in terms of all the different molecules that are involved in function. And from the standpoint of, number one, identifying pathogens and foreign objects and cancer cells and attacking them, while not attacking self. And in the case of autoimmune disease, there's a dysfunction in which the immune system does attack self. And IBD is one manifestation of this failure. And so you can see that in this case, the infliximab is helpful compared with what there was before. Does anyone know what they used to treat IBD before?
What is a steroid?
Something like
Yeah,
sulfasalazine.
Right. Yeah, steroids, which can be pretty effective as anti-inflammatory. But with something like an autoimmune disease where you have to have sort of chronic therapy, the adverse effects associated with steroid therapy are profound. Can anyone give me an example of an adverse effect associated with prolonged
Cushing syndrome?
Say again?
Cushing syndrome.
Okay. Could you describe that just a little bit? What kind of syndrome again?
Cushing, like a moon phase, and they have a lot of like.
Right. So changes in fat distribution. You could also have other endocrine problems. You could have psychiatric problems. You can have essentially a decrease in bone density leading to osteoporosis, whole range of bad stuff. So that wasn't very good. And then there was just sort of salicylates, which of course have a whole range of adverse effects themselves and are not especially effective. So the use of these antibodies are really a step up, although they are not without their own adverse effects. So let's move on here a little bit, and we'll take a look at some of the things that the use of infliximab does. So just looking at the top of the table here, it downregulates inflammatory cytokines. That's a good thing. It can inhibit apoptosis, although it talks about apoptosis here. I think it's regulating apoptosis of inflammatory cells. It induces this regulatory macrophage, which is sort of a good thing, because that macrophage then can prevent damage associated with other inflammatory cells. It protects against loss of the intestinal cells related to a barrier. Loss of that barrier, aside from infection, can also lead to, once again, interactions with other inflammatory proteins. These are other inflammatory proteins that are downregulated, protects endothelial function. Angiogenesis, what's up with that? What is angiogenesis?
Formation of vessels.
Angiogenesis is the process of the production of new blood vessels. That's commonly seen in chronic inflammation, and it's also seen in tumor growth, solid tumor growth. What are MMPs?
Matrix metalloproteinase.
Right, which is also involved in the inflammatory process. By the way, which metal is involved in that protein?
Zinc, I think.
Correct. That's absolutely right. Promotes mucosal healing, inhibits NF-kappa B signaling that we just talked about, but it promotes MAP kinase signaling. So is that a good thing? Maybe, not necessarily so. Because MAP kinase signaling can also be involved in inflammatory response. And so you can see here that this necrosis factor inhibitor inhibits inflammatory cytokines, which is good.
Sir,
Yes,
I have a question.
sure.
So in the start, it was written that TNF, with the help of TNF receptor 1,
Right.
activates both the intracellular pathways, the MAP kinase and both, like NF-kappa B. But
Right.
over here in the table, with the use of infliximab is basically inhibition of TNF alpha. So how can it's possible, like how it's like one of the pathway can be promoted and other can be inhibited?
What do you think?
Like I cannot understand this point. Like it's the same, like TNF alpha is basically activating both the pathways, but inhibition of TNF alpha causes, it's written in the table that promotion of MAP kinase and inhibition of NF-kappa beta, kappa B.
I think that's addressed farther on in the paper. So we'll talk about that a little more when we get there. That's okay.
Okay.
So this granulocyte macrophage colony stimulating factor is linked to inflammatory bowel disease, which then is also associated with inflammation. And it's increased in Crohn's disease. But they're decreased after infliximab, indicating a role of infliximab in controlling the inflammation that's linked to this granulocyte colony stimulating factor. What is this GM-CSF, and what does that do?
Formation of granulocytes.
And just quickly, what's a granulocyte? Can you identify some?
Neutrophils.
Neutrophils, basophils, eosinophils.
Yeah.
Right. Okay. All involved with cellular immunity. Induces immune cell apoptosis, right? So that's listed in the table. Through signaling through transmembrane TNF. By the way, what is this transmembrane? What forms does TNF actually take?
There are
So
two
that's
forms.
There's soluble and there's the membrane bound.
There's both membrane bound and soluble trimer, right? Homotrimer. And so either one, whether it's membrane bound or soluble, can bind to its receptor, which then triggers all the responses that we've been looking at least from the standpoint of NF-kappa B and perhaps MAP kinase. But they haven't really gone into the connection there quite as well. Here they note the drug sustained mucosal T apoptosis in Crohn's disease. By the way back in the Pleistocene era when I was a graduate student, my advisor told me that this word, apoptosis, is actually pronounced apoptosis, so the first P is silent. Have any of you ever heard that? No? Well, he's from Boston, and maybe that's just a Boston thing. I don't know. But forgive me if I don't pronounce the first P. No, the second P, apoptosis as opposed to apoptosis. Okay. So they mention here that irritable bowel disease is characterized by an imbalance in T helper cell responses with dominance of Th1 and Th17, while there's a deficiency in Th2 and T regulatory responses, which then is associated with IBD, and apparently with the infliximab, you can help to restore the balance by increasing Th2. And then Th regulatory cell, which once again by restoring the regulatory response of this one, let's see which one is that, T cell Treg, can restore more normal function. So you see increased Treg apoptosis in irritable bowel disease, and insufficient compensation for chronically activated T lymphocytes. Okay. Protects epithelial cells, intestinal, from apoptosis. So after 4 weeks, there's a decreased intestinal permeability in Crohn's disease. Expression of leukocyte endothelial cell adhesion molecules. And we looked at this a little bit on Tuesday. So during irritable bowel disease, you have an upregulation of vascular cell adhesion molecules, BCAM1, and intercellular adhesion molecules. After infliximab infusion, the upregulated leukocyte cell adhesion molecules were restored, I think, more to normal in parallel with the disappearance of inflammatory cells. So the bottom line is that the drug regulates immune cell infiltration by controlling leukocyte endothelial cell adhesion molecules. These molecules have to do or play a role in infiltration of immune cells into the inflammatory tissue. Angiogenesis, shown to be critical component of neoplastic and chronic inflammatory disorders. So they say another effect of infliximab is the downregulation of mucosal angiogenesis and restraining the production of vascular endothelial growth factor. Then they go into regulating inhibition of metalloproteinases. So it seems like it has a host of anti-inflammatory properties. The question is, are all of these linked to tissue necrosis factor? Or does it do other things? What do you think?
So far, it looks like everything is either directly or indirectly related to tissue necrosis factor or inflammation. Like the matrix metalloproteinase function, it's related to TNF. But improving mucosal healing, it's indirectly related to TNF. Like if the inflammatory markers and their response goes down, the mucosal healing will be promoted. So I think TNF is the main target for this drug.
Okay. But everything seems to be connected and influencing others. We were talking a little bit earlier about MAP kinase. And here, another group found that infliximab enhances the activity and expression of MAP kinases during IBD. MAP kinases are a key signaling pathway that cooperate in the orchestration of inflammatory responses and extensive crosstalk with other inflammatory pathways such as NF-kappa B, So maybe you can activate NF-kappa B via MAP kinase as opposed to just TNF. And then there's the Janus kinase, C-Jun N-terminal kinases, stress-activated protein kinases. So all of these transduction pathways are activated in inflammatory bowel disease. And it says in vivo inhibition of TNF by infliximab resulted in a significant transient increase in P38 activity during the first 48 hours after infusion. Significant activation was also observed in myelomonocytic cells. So this sort of indicates then that is TNF the major player here, or it may be a major player, but it may not be the only player. Is that fair?
Yeah.
Okay. Okay, it's noted in clinical trials for irritable bowel disease that the response to infliximab treatment is only about 60%. And only about 30% of responders had a sustained remission throughout 54 weeks. And this lends itself to why some people are benefited by the drug and other patients are not. And I think it goes down to this: genetic predictors of infliximab through essentially polymorphisms of particular genes. And they're sort of pointing at caspase 9. Remember, caspase 9 is actually an early on molecule in the cascade leading to apoptosis. And it's linked especially to luminal and fistulizing Crohn's disease, essentially the wearing away the lining of the GI tract. And this particular genotype, the strongest association with a non-response, while concomitant or captoprine azathioprine therapy was linked with the effect of unfavorable genotypes in Crohn's disease. So apparently, the issue with infliximab not being especially effective across the board is linked to genetic polymorphisms that somehow have an impact. Polymorphisms in tissue tumor necrosis factor, the tumor necrosis factor receptor, right? From the standpoint of the mechanism of action of the infliximab, how could these polymorphisms have an impact on the drug? What do you think?
So, Dr. Johnson, polymorphism in FAS ligand and caspase 9 may possibly suppress the apoptosis of T cells, like the activated T.
Mm-hmm.
That kind of shows the significance of apoptosis here.
Right. But my thought is this, and I'm just speculating here, and I'd appreciate either you folks telling me I am off base or if I'm right on. If you have a polymorphism in either the TNF protein or the TNF receptor protein, could that have an impact on the binding of the infliximab antibody to these proteins? So if the antibody doesn't bind, you're not going to see the therapeutic effect. Is that fair?
Yes.
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 Okay.
But there are also other. It depends actually on the polymorphism because there are certain types that will either enhance affinity to binding or decrease binding. It depends on what happens to the proteins.
Right. The polymorphism would have to happen at the target binding site of the antibody for the protein. Is that correct?
Yeah, for this case, yes.
Right. So polymorphisms in other portions of the protein may not affect the binding of the antibody, but if it's in the target site of the antibody, maybe it would affect binding.
Yeah, that seems possible. Yeah.
Johnson, adding to Melanie's, like the gain of function, there's something called as gain of function polymorphism as well, where probably the polymorphism enhances binding of antibody to the receptor.
Right. Could a gain of function polymorphism cause the protein, the TNF, to bind to its receptor even if an antibody is bound to it? You see what I mean?
Mhm.
So, the TNF has got to have a portion of the molecule has to be the binding site for the receptor. And if the antibody targets the binding site of the receptor, it won't bind to the receptor. If, however, the binding site of the receptor will bind with even higher affinity for the receptor than otherwise, and then maybe the antibody isn't binding to that site, you can have an increase in the NF-kappa B anyway. And then they talk about different homozygous carriers of inflammatory bowel disease risk increasing the IL-23 interleukin 23 receptor variants were more likely to respond than homozygous carriers of risk decreasing IL-23. So once again, this could be downstream with interleukin 23. I don't even know where that comes into it. What is interleukin 23? Anyone look that up?
So, Dr. Johnson, it is cytokine that is that increases during Th17 response, and mostly related to Th17 is mostly pro-inflammatory, like even more inflammatory than the cytotoxic T cells.
Okay. Now, I'm just going to drop down here to side effects. Infusion reactions. What is an infusion reaction? A delayed that's an allergic response, isn't it?
So at the site of injection, maybe.
Sorry, Vivek, go ahead.
No, go ahead.
Especially when a drug is given intravenously, so patient responded like kind of violently after the treatment. It
Mhm.
happens with insulin also.
Ah, okay. Especially, I would think, does that happen more with insulin from animal sources as opposed to human transgenic sources? I think so. Leukopenia, what is that?
Decrease in leukocyte count.
Right. Why do you think that would happen?
Because in the table 1, it showed that it causes leukocyte apoptosis.
Okay. And then, of course, if you're interfering with immune function, increased risk of infection, antibody development against the chimeric I would assume the murine portion of the chimera, and then increased risk of malignancy, because I mean, a major role of the immune system is to identify and destroy neoplastic cells. It says here, severe acute infusion reactions strongly associated with the development of anti-infliximab IgG antibody, and that the risk was particularly high with the second infusion in a treatment series. And what about this? What is this? Gamma delta T cell expansion in vivo, indicating the potential risk of developing this. Is that a delta? Gamma delta T cell lymphomas? Other autoimmune diseases. What's up with that? Why would the use of this drug induce other autoimmune diseases? Such as psoriasis. Well, maybe you're activating other components of the immune system. We also mentioned MAP kinase, and maybe that can trigger an inappropriate immune response to some protein in the integumentary, the skin system.
Dr. Johnson, what the reference they cited in this paper, so in that paper it says that TNF alpha kind of suppresses the production of interferon alpha. So infliximab inhibits TNF alpha activity, interferon alpha levels goes up, which is directly related to psoriasis. So that is, yeah.
Very good. Very good. I'm impressed. Very good. Thank you. So, their conclusion, TNF is a key pro-inflammatory cytokine during the development of IBD, plays a pivotal role in modulating the mucosal immune response, including regulating immune cell proliferation, upregulating adhesion molecule expression, and inducing, I'm going to use the pronunciation, apoptosis of intestinal epithelial cells. However, the biological action of the drug on immune diseases is far from being well explored. T helper 17 cells have been shown to participate in progress of IBD, but so far no evidence is available about the impact of infliximab on T helper 17 cells during the process of inflammatory bowel disease. A recent study found that interleukin 15 and its soluble receptor might mediate the response to infliximab in patients with Crohn's disease. How could that happen? If it's a monoclonal antibody against TNF, what would IL-15 have to do with that?
IL-15.
The other thing here, they comment, infliximab is useful for other immune diseases such as rheumatoid arthritis, and that immune response is typically not associated or characterized by TNF. So infliximab is doing other things, but once again, if it's an antibody against TNF, how can it react with other molecules? In this way, we believe infliximab is supposed to play biological actions beyond neutralizing TNF production and counteracting its activity. Now, this paper was published in 10 years ago. So they probably know more about it now than they did then. Also, this was one of the first drugs of this type to be approved and developed. It'd be interesting to see if more recent drugs are superior to this one or not. Any thoughts on that?
Sir, I was just thinking about it that maybe the difference in the outcome of or the result of infliximab in IBD patient can be due to variation in the role of TNF alpha in the individuals of IBD.
That makes sense. That does make sense. Well, this is worth pursuing further. I don't know if I'm going to be with you next Tuesday or whether Dr. Kavanaugh will be teaching. I'll find out, and if it's me, I'll send you another paper for us to review. Hope you all have a good weekend. It's supposed to rain on Saturday again, so that's a bummer. But you graduate students never leave the labs anyway, right? So you don't have time to go outside. You got to be generating data. But in any case, if I do have a chance to see you again on Tuesday, I look forward to it. You all take care.
Thank you.
Good.